Hi, everybody. Good morning. Thank you very much for coming to this session on evolutionary rescue. We're very excited to have a great lineup of speakers and talks today. Hope you enjoy, and we'll get to it quickly. Our first speaker is Danna Gifford from the University of Manchester. Thank you.
Thanks, Matt, for the introduction. Well done, everybody, for making it to the morning of the last day. So I'm very happy to be in this Evolutionary Rescue Symposium. Our group works on antimicrobial resistance. So in general, we're trying to prevent evolutionary rescue from happening, although sometimes we don't, and sometimes when that happens, we learn some fun things along the way. So one area that we've been focusing on in the last couple of years is combination therapy. And so if that's not a term you're familiar with, it's really quite simple. In contrast to the situation where you give one drug and resistance to that drug readily evolves, or a second drug on its own, to which, again, resistance readily evolves, the idea here is that you use two drugs together, so A drug plus B drug together. And the idea with this is that it's supposed to prevent evolutionary rescue from happening through a couple of mechanisms. One, the first thing is getting resistance to both drugs through a simultaneous mutational event is supposed to be too rare for that to happen, just because mutations individually are quite rare. So if you combine them together, that's a really rare event. So we kind of avoid this pathway from happening directly from a sensitive strain to a double resistant strain. The other thing that's supposed to be prevented from happening is this sort of sequential acquisition of resistance. So imagine you get first resistance to drug A and then a second mutation to drug B, or the opposite situation, first B and then drug A. This should be prevented by the inhibitory actions of the other drug. So drug A inhibits resistance to B, inhibits things that are only resistant to B. Similarly, drug B inhibits things that are only resistant to A, and therefore you don't get the sequential evolution of resistance. That should be true in theory, but it sometimes happens nevertheless. So we want to know this black box here. How does that happen, and what happens along the way when that goes on? So to answer this question, we used a technique called experimental evolution. Hopefully some of you have heard about that already, but that's where we take microbes in the lab and evolve them to be resistant to antibiotics on purpose. I swear we do this in laboratory strains for the most part that can't then escape out into the wild, so it's just kind of a toy system. So our favorite one that we use quite often is called E. coli K12. This is a particular strain of that that we know quite a lot about the genetics of. And we can do things like expose this to antibiotics under controlled conditions. In this case, we used two drugs, one called rifampicin, one called nalidixic acid. And the choice of these was because we know a lot about how things evolve resistance to these. So we did these two drugs on their own and then in combination. One thing we did a bit different was we spiked in things with an increase in mutation rate due to a defect in their DNA mismatch repair system. We put these in at frequencies around what you would see in some clinical infections. So these have a higher mutation rate than the wild type by about 80 times. Finally, we raised the drug concentrations and tracked resistance phenotypes over time, and this is where the evolutionary rescue aspect comes in. If they haven't achieved resistance by sort of the time we hit the full inhibitory concentration, then they should go extinct. So in my talk today, I'm only going to focus on the no antibiotic and the combination treatment just in the aspect of time. So we tracked resistance or different phenotypes through time, either no resistance, single resistance to rifampicin, to nalidixic acid, or another case where we have resistance to both single drugs in separate individuals, but not in the same individual, versus this bright pink double resistance. So when I say rescue, look for these bright pink double resistant bars. So the first case we looked at, when we had no mutators, so nothing with an increased mutation rate, we observed no rescue. Basically, things bumbled along without resistance in both the antibiotic-free control and the combination, and by the end, all of the populations here had gone extinct in the combination environment. In contrast, when we spike in as few as 5% mutators, we start seeing rescue happening. So we get these bright pink bars happening here, indicating that they were able to find these two mutations. And crucially, in the drug-free environment, there was no rescue or double resistance going on. We only ever saw the single drug resistant individuals. So this suggests that actually it's the environmental change that's helping this go on. And as you might expect, if you add in more mutators, you get more rescue. So the bars get bigger here when you have 30% mutators. So this was, a nice experimental system, and we complemented this with some stochastic simulations. And if you saw Ernesto Barrios' talk earlier this week, he was really the brains behind that. And quite unusually, we got a fairly good concordance between our experimental system and our basic Lotka-Volterra-based simulation model. So we were quite happy with that. We figured, okay, we've understood that at the end, mutators facilitate resistance evolution, and that's all we need to do. So, of course, we submit things for publication, and we get reviews back saying, you haven't sequenced anything. We said, well, we don't really need to because we know how the resistance evolves in this system. It's not really worth our time. But in the end, we gave into the comments. And I'm really glad that we did because we found something really unexpected that our model didn't account for, and something that I think is really important to think about when we employ these evolutionary motivated interventions into these types of systems. So right, we took some of the rescued populations and we sequenced their genomes to look for the mutations that happened. So we took 30 survivors from each of those different, well, we didn't have any survivors when there were no mutators, but in each of the 3 different levels of mutator frequencies. So as we expected, we found mutations in those 2 canonical targets of resistance, the RPOB gene, which is to do with RNA synthesis essentially, and GyrA, which is related to DNA synthesis. So that was quite expected. And we did see some diversity in terms of the exact mutations that occurred. We recovered the hotspot mutation that often happens in the species, which was nice to see. So that all made sense. But when we looked at the rest of the genome, that's when we really kind of got excited with this data, I think. So in addition to those 2 key rescue mutations, we saw a striking number of mutations per isolate in addition to those rescue mutations. Now, it happened over the course of a week-long experiment, which I forgot to mention. What was really striking when we look at this distribution of mutational frequency, so this is the number of mutations per isolate, we see some pretty extreme outliers over here. So this is really kind of a lot of mutations if you're not used to looking at this data. We wouldn't really expect to see 160 mutations after 1 week's experiment. So we again looked at a per isolate characterization of the mutations that happen, and we saw some striking outliers that were going on here. So we decided to follow this up by actually measuring the mutation rates of these populations that had rescued. We found that in 5 out of 30 of those isolates we sequenced, we had additional mutations in different DNA replication fidelity systems, including in one strain where we observed an increase in the mutation rate up to 1,000-fold higher than the wild type from its initial 80-fold higher mutation rate. So this was due to a mutation in the core DNA polymerase, which is supposed to be a really high-fidelity DNA polymerase, and it happened in the error correction subunit. So that was really quite surprising to us. We didn't expect to see that over such a short time scale. So we've now taken these 3 isolates, so the wild type, the original mutator, and now our high mutator, and we asked the next question, which is, does this now facilitate rescue to a new environment? If you put them in another challenging situation where you increase the drug concentration, do we observe the same process happening for different conditions, and do these also have an advantage in different environments? So we switched to using an antibiotic now that is actually quite relevant for the treatment of E. coli infections. It's a first-line treatment for urinary tract infections, which E. coli is the #1 causative agent of, and actually has a low prevalence of resistance globally. So despite being in use since the 1950s, it's kind of an outlier in terms of its ability to suppress resistance. This is showing just different differences in usage versus resistance in the UK, where there was a switch in 2015 to this being the first-line drug, and we didn't see an increase in resistance. So that's quite special as far as antibiotics go. Crucially, like the combination treatment, we need 2 mutations for full resistance to be observed to this drug. So we thought maybe this is another case where we might see mutators evolving in these genetic backgrounds. So we did the same thing. We ramped the drug concentration over time, and again, we saw striking differences in the proportions surviving, although in this case, the wild type was able to survive a little bit under the conditions, which we changed slightly because this was a student project. We needed to get some results, so we made it a little bit nicer for the bacteria. But we still observe this difference in the survival probability, increasing with increasing mutation rate. Again, we saw really a lot of mutations. This time, 300 and I think that says 75 mutations on average in the course of a week-long experiment. That's really quite a lot of mutations in that lineage that gained the now 1,000-fold higher mutation rate. But in this case, we didn't see any obvious outliers where we had a jump in the number of mutations relative to other things from the same treatment. So we don't think that mutation rates actually evolved further here. We need to do more work to figure out why these conditions didn't favor that and the other ones did. So actually, That said, though, we did observe, just because of the really high mutation rate, that there were some additional defects in some DNA replication fidelity systems in the already high mutation rate lineage, but we suspect that they're just there because the mutation rates are really high. They probably don't have any functional significance. What we did notice, though, is that even though the rescue probabilities, were slightly higher and we didn't observe increased mutation rates, we did observe that the higher mutation rates resulted in better resistance mutations. Even though we're in a evolutionary rescue kind of regime, we did notice that there were key differences in what types of mutations the three different strains got. So the wild type mutations got sort of blunt force mutations where they either had large deletions or like big disruptions to the key genes involved, meaning that these genes have probably completely lost their function. In contrast, in the two mutator lineages, we saw single nucleotide polymorphisms, which were a little bit more subtle. So in the sort of intermediate mutation rate, or here it says low mutation rate strain, we saw sort of gain of stop codons in the coding sequence. In the high mutator, though, we saw specific residues mutated that are key for the function involved in this protein. So I don't have time to go into that. The take home from this, though, is that more mutations means more specialized resistance in this case. We've also started to look into these mutations in clinical isolates, and we're starting to maybe see some putative mutator alleles co-occurring with resistance to this antibiotic. We need to do more work on this to really shore up that finding, though. So to wrap up what I'm saying, I think we think a lot about how mutation rate facilitates evolutionary rescue. But one thing, at least for bacteria, I think we need to think about as well is the other way around. Does evolutionary rescue facilitate mutation rate evolution? Some questions I think this has thrown up is evolutionary rescue a driver of mutation rate evolution in these bacterial populations? Is there a minimum mutation rate to potentiate this mutator evolution? We never saw it happen in the wild type background, but we did see additional mutation rate evolution in the already mutator background. And so this is just something I drew. This is not a model, but you could imagine some regime where you have extinction at high rates of environmental change and low mutation rate, rescue all the time at low rates of change and high mutational supply, but then some sort of regime in the middle where you get rescue and a mutator allele kind of sweeping to fixation together. I don't know what the shape of this is going to be. It could be something like that or something like that. If anyone knows, please tell me so I can stop thinking about it. Ultimately, there might also be diminishing returns or mutational meltdown. There's probably an upper limit to how much things can mutate and rescue. And finally, does rescue beget rescue? And this is really important, I think, in this resistance context, where if you get resistance to one drug through this process, are you then more likely to evolve resistance to further drugs? Ultimately, again, I think we, as I said before, need to consider the unintended consequences of evolution-based interventions. We had a good model. It fit our experimental data, but we were missing some key components there, and I think we need to think about those things before we do things, to real people. So with that, I'd just like to thank the people who did the work, so experimental and theoretical colleagues, and my research group as well, Merman. We're a great group. If you want to do postdocs and PhDs, come and talk to me. Don't have to work with me. It's about 12 PIs, so happy to. I think I've got some time to take any questions. So it's evolved previously at the same exact locus in that one gene. It's known to experimental evolution, slightly different SNP, but yeah, that one position is in the exo-1 polymerase section that's important for the error correcting thing. And yeah, scarily we're seeing mutations in that same gene in the clinical isolates, but I need to confirm whether it's actually having a phenotypic effect, yeah. Yeah, thank you.
One in the back. When you say the mutations are better, is it just, so I know you said they're better because of resistance, but what about like compensation or like the fitness of them, especially in the absence of antibiotics?
Yeah, we have that data. I didn't have time to make a nice plot out of it, but yeah, that's basically what I'm trying to say is, it might affect its ability to bind the antibiotic and not its normal ability, which is to reduce nitrogen groups or something like that, yeah. Do I have
So I found it interesting that you already had a mutator and there was evolution of more mutations. Do you need a mutator to have evolution of another mutator? Or?
That's what I want to find out. I have seen these they're now called by others multi-locus mutators, so other people have observed this as well, and I think there probably is a minimum mutation rate for which you need to see this second mutator allele arise. But yeah, I want to do that with simulations, and if anyone wants to do theory, then yeah, happy to do that too.
Right, so I'm going to try to do this without seeing them, which is sort of fun. So we often frame consideration of a population's ability to track environmental change in terms of a phenotype that is related to fitness. So we have here sort of a cartoon of some sort of fitness landscape, and we've got a phenotype that at this particular environment maximizes fitness. But of course, when we think about evolutionary rescue and these sorts of questions, we think of a change in time in the environment that leads to some sort of new fitness regime. And so now we've got a different phenotype optimum, labeled here theta. And if you have the same mean phenotype as before, you're at low fitness. And that low fitness, we have to show this once at least. So here it is. That low fitness is what causes the demographic decline that is linked to the environmental change and will eventually lead to population extinction unless something happens. And the thing that can happen is evolutionary rescue, right? Adaptation. So in this case, it would be an evolution of a phenotype. But it's this low population size that makes us worry about the kind of stochastic effects that can lead to environmental loss. So my lab, this is also related to the linked consideration of spatial structure. So here we might imagine some different kinds of habitats, and maybe in these types of habitats here, if the population has a particular mean phenotype, it's at the optimum, and so it has high fitness. But in another area of habitat, say this outer ring, there's a different optimal phenotype, theta 2 instead of theta 1, and now if the population has that same mean phenotype, it's at low fitness. Of course, it can rescue itself by evolving towards that optimum, but this is complicated by gene flow, right? So this is a pattern of maladaptation in space that is analogous to that same maladaptation in time that we just talked about. My lab's really interested in how reproductive mode affects the ability of different kinds of populations to track environmental change and to adapt to new habitats. And the reproductive mode we've been considering is partial clonality, which includes both sexual and asexual reproduction. And asexual or clonal reproduction, there's a huge range of it. Things like vegetative growth and spread, like this is a, I think this is Vitex rotundifolia, which is a invasive plant. This is on a beach in Hawaii where it doesn't belong, and spreading vegetatively. But that same sort of vegetative growth and fragmentation can lead to clonal reproduction in things like marine animals, like these corals. Obviously, there's things like this sort of poster child here for clonality is hydra, it's budding. But then there's things that are more less obvious. This is a grass, Poa bulbosa, and it's producing dispersal things. They're not seeds. They're called bulbules, and they're completely asexual. So there's a wide variety. We're also really interested because we see this relationship between clonal reproduction and invasive spread really often. Here's two sort of classic examples. Here's invasive seaweed, Gracilaria. And over here, this looks like a field, but it's not a field. It's a body of water completely covered with what might be a single clone of this aquatic fern, Salvinia molesta, which spreads vegetatively. So we've been developing models that look at phenotypic evolution with stage-structured life histories. This lets us look at complex life histories and include both sexual and asexual reproduction. We've based it on work by Barfield et al. In our own work, and in sort of a classic quantitative genetics framework, we look at a phenotypic trait, and we partition that trait into the additive genetic factors. So under sexual reproduction in random mating populations, it's only that additive genetic factor that gets passed on to offspring. Stop gesturing. And then, I'm Cuban, so it's really hard. Okay, and then E basically is everything else. So non-additive genetic factors and environmental factors that determine phenotype. That non-additive genetic factor, though, includes things like dominance interactions and epistatic interactions. If you're reproducing clonally in a diploid, you're passing on all of that. So some of this E gets passed on under clonal reproduction. We develop recursions for the number of individuals for each stage, for the joint probability density function of phenotype and genotype, and I am going to not show any of those details today. But I am going to talk briefly about a parameter that we define, rho. This gives the association between E, that non-additive genetic plus environmental factor, in parents and their clonal offspring. Rho is 0 for sexual reproduction and random mating. Rho might be close to 0 for some types of asexual reproduction, depending on the genetic architecture of the phenotypic trait or the type of clonal reproduction. But it might be closer to 1, again, depending on the type of phenotypic trait and how much of the parental tissue ends up being part of the offspring. So I'm going to give you just a few examples from our work. Here's one for a simple life history that combines both clonal and sexual reproduction, where we have selection acting on survival probability. In a really classic case where basically your ability to survive depends on how far your phenotype is from the optimum theta. We also track the amount of clonal reproduction as a proportion. It's the proportion of the total sexual fecundity plus clonal reproduction that's clonal. And we develop difference equations for the phenotype and genotype. I'm going to show you some of the numerical analytical results here. So again, this simple case, we're going to contrast no clonality with a case where R sub C multiplied by rho is 0.5. So at the top here is the value of the genotype and phenotype. We start this numerical exploration with a mean phenotype and genotype of 0, and the optimum is 3. And you'll see that natural selection works, yay. They evolve to 3. And you might see, this is a little faint here, but the no clonality, purely sexual cases, the dotted line, it gets there first. So clonality slows the approach to the optimum. But it also does something else. It separates the phenotype and the genotype. And you see that we see this initial increase of the phenotypic value towards that optimum. And that initial growth lets the population get out of that decrease in fitness quicker. So down here's the population size. So if they start off with some number, and of course they're really maladaptive, they have a phenotype of 0, and they plunge. But the sexual population, now the solid line, dips much further and takes longer to recover than the clonal one. Now this is analytical work of a deterministic model. There's no stochasticity, no one's going extinct, and yet we start to worry, oh, this looks like where we might go extinct. So let's add stochasticity. So we did that by looking at individual-based model simulations. There's a lot of details I'm not going to go into. I'm just going to say that we did consider 2 different types of changes in the optimum phenotype, a single sharp one-step change and continuous linear change, but this is a 12-minute talk, so today we're just going to talk about a one-step change. And here are the results for the simulations. This is the probability of persistence, 1 minus the probability of extinction, as a function of the size of the step change, how big of a shift in the phenotypic optimum there is. And you can see, I've drawn arrows to help us, that as we increase the amount of clonal reproduction, or the proportion of clonal reproduction, or that association parameter rho, we increase the probability of persistence. So this means that this matches our intuition from the analytical model that clonality is actually helping the populations through this demographic advantage. So this is what I just said, and the reason for this, if we think about how you survive a step change, a sudden step change, you're depending on standing variation, and clonal reproduction allows more of the good phenotype that happens to be there to be passed on to get you through that window. That's that demographic advantage. If we then think about spatial structure, let's think of the very simplest case. We've got 2 demes now, and they each have a different optimum. So we might imagine a theta-1 and a theta-2. And we imagine a model where first we have migration of juveniles, then we have selection on survival to adulthood, again, as a function of how far away the phenotype is from that optimum. And there's 2 different optima. And then we have reproduction that's both clonal and sexual. Now we have analytical results, but I'm just going to show you the results from the simulations for time. So this is under symmetric migration between the 2 demes. And what we see is a recapitulation of what we saw for the change in time, where as we increase clonal reproduction or as we increase the association parameter, the probability that deme 2 adapts increases. Delta theta here is the difference in optimum between the 2 demes. Now, that's until you get to completely asexual reproduction. I highlighted in red the line for when all of the reproduction is clonal, or the association parameter is 1. And you see this one goes down just a little, this one goes down quite a bit. You need a small amount of sexual reproduction for this to work. That's results that have been found previously as well. But sometimes you have asymmetry, and Ophelia's in the audience, so I have to not mess this up. Okay, so one of the issues, though, is we were just considering an symmetric case, but not all spatial cases are symmetric, and dispersal asymmetry biases natural selection towards the source habitat. So on the top here we have a measure of fitness, so we have 2 demes with 2 different optima, just as before. Under symmetric migration, both of the deems are able to adapt. They're off a bit from their optimum, but they're both able to adapt and maintain populations. But if we have very asymmetric migration, what can happen is deem 1 can act as a source into that sink deem, and it can keep deem 2 from adapting. It'll have a low population size, and it can go extinct. This is sometimes called migrational meltdown. So we explored this, and I'm just going to show you one result, again, from simulations, although we also have analytical results showing the same thing. So here we modeled this asymmetry by a difference in the carrying capacity of the two deems, so the size. So we have a big deem and a little deem. When the little deem is empty, we see the same thing as before. Increasing clonality makes it easier for that deem to adapt. But when the little deem, the sink deem, is full and at its optimum, now there's a range of values of differences between the two deems where the population goes extinct. This is migrational meltdown. This is the line with the solid circles for completely sexual. As we increase clonality, that range of parameters decreases and eventually completely goes away. So we see that greater amounts of clonality permit adaptation under a greater spatial selective difference that parallels the finding for that one-step change in time. So we're seeing the same sort of rescue in both time and space. And for migrational meltdown, partial clonality reduces the cases where dispersal can cause habitat loss. We think that maybe this is a possible explanation for the success of many partially clonal organisms in invading new habitats. Of course, that's going to complement traditional explanations that are based on things like allele effects, such as reproductive assurance. There was no issues with that in this case, the way we modeled it. So we were just isolating this. And I don't know how fast I talked, but pretty fast. So I'd like to acknowledge our funding sources, and I'll mention that this is work carried out by Bob Holt and Mike Barfield. Thank you very much.
We have a few minutes for questions. Yep, in the back.
Hi, thanks, Dr. Maria. I have a question about the probability of extinction that you may have been on there, but I'm kind of in the back. So with the probability of extinction, is that including or taking into account of all individuals, or are we saying like probability of extinction, like say of a stem?
So the way So for these simulations, the way we did it, we have individuals, and they are hermaphroditic, so they can, as long as you've got one, you've got a mate. So they don't have to worry about finding a mate. And the simulations, I think we did 400 runs, or either 400 or 500 runs for each set of parameters. And basically, you're extinct when you have nobody, when you go to 0. Because as long as you've got one, you're not extinct yet. Yeah, so this, like a clonal lineage. So I should mention this, you've brought up a really good point. These organisms can either reproduce clonally or sexually each generation. So we don't have lineages that are only producing clonally and lineages that are only reproducing sexually. That would look a little different. That's a good point. Thank you. Yes?
I was wondering, in your exploration, do you assume that you can vary parametrically, independently, the amount of clonality and the amount of standing variance available?
So that is an issue as well. So in the simulations, what we did to just look at the effects of these parameters, we start when in these it's all the step change. That we start with all of the populations having the same amount of standing genetic variation, and so we're basically looking at the loss of it. You're right to say that you might consider a population that has more clonality all the way along would have less standing genetic variation at the beginning. So we're just looking at like modifying this one parameter, but you're right. We could also do that. Thank you. Yeah.
Hello, everybody. So my name is Julien, and today I'm going to present some of the work I did during my first year of PhD. So I'm going to talk about evolutionary rescue and complex life cycles. As you all know, many species are threatened by extinctions, and especially due to the increase of the temperatures. And some of the most threatened ones are the endemic species. And Centaurea corambosa is an Asteraceae micro-endemic from the southern France. And the species has been surveyed since 1994. And over the monitoring period, as you can see here, all the 6 natural populations are declining, so the species is threatened. And because Centaurea corambosa has really poor capacity of migration, the best way to avoid extinction in the long term is to adapt genetically to the new climate. And in this case, we would observe evolutionary rescue. And as many of you know, one of the most important parameters favoring rescue is the amount of heritable genetic variance in the population. The higher it is, the higher the speed of evolution, and thus the higher the probability of rescue. So if this amount is high enough, the population is saved. If there is too little genetic variance, the population goes extinct. Most theoretical work on evolutionary rescue considered homogeneous populations. In reality, species have complex life cycles. So you can see here the Centaurea corambosa example. So the species form structured populations in which individuals transit between 3 different stages. So the seedlings, the rosette, and the flowering plants. And these stages differ in their sensitivity to climate variables. So for example, here the survival of the seedlings is strongly affected by the temperature during the summer. And another property of this complex life cycle is that the different life history traits, so survival, fecundity, do not have the same contribution to the demography, to the population growth rate, and it's traditionally measured by the elasticity of the life history trait. So you can see here that the 4 life history traits of the Centaurea corambosa life cycle differ in their sensitivity. And as a consequence, the evolution of these different life history traits do not have the same impact on the demography of the population. And however, most of the few studies on evolutionary rescue in structured populations investigated the impact of the evolution of a single life history trait, most often the survival of the juvenile, on the probability of rescue. So the aim of this study is to understand how the genetic variance of different life history traits influences evolutionary rescue probability. So to answer this question, I used a modeling approach, and I assume that each life history trait of the life cycle, so here you have an example with the seedling survival, each life history trait of the life cycle of Centaurea corambosa is associated with a latent phenotypic trait Z and maximized for a given value of this trait Z. And using the data from the demographic surveys, I was able to parameterize the model, so to identify the maximal value of each life history trait and also the current average value of each life history trait in the natural populations. And so I was able to compute the maladaptation on the Z trait. So all my recursions start at the same starting point. It means at the current demographic situation of the populations. So all the populations are declining. And so I used classical quantitative genetics assumptions. So in reality, my latent phenotypic trait is a quantitative trait. And I assume that so the phenotypic distribution and the distribution of the Gaussian of the breeding values to be Gaussian, and I also assume that the genetic and phenotypic variance stay constant across life and time. So I used the model developed by Barfield and colleagues to model the evolution of each life history trait through the evolution of each trait Z. So I explored 2 main scenarios. The first one is an univariate scenario where a single life history trait is genetically variable and can evolve. So in this scenario, I attributed genetic variance to a single life history trait and set the other ones to their average value. And so I ran several recursions of 500 generations long, and I just changed the initial variance, genetic variance on the considered life history trait. So I had many trajectories, and for each trajectory, I identified the minimal population size reached during the 500 generations. And because I work with structured populations, all the individuals do not have the same contribution to population growth rate. So I weighted population size by reproductive values. So I was working with minimal total reproductive values instead of minimal population size. So just before my results, I show you some analytical predictions that we derived. So again, in this univariate case, the change of the mean life of a given mean life history trait is approximately equal to the product of its additive genetic variance and of its elasticity. So you have here an example, again, for the seedling survival. So we expect the traits with high elasticity to evolve faster, and so the minimal total reproductive value to be higher when these traits evolve. So now I'm going to show you my results, again, when a single life history trait is genetically variable at the time. So on the y-axis, on the following figures, you have the minimal total reproductive value I told you about before. And on the x-axis, you have the additive genetic variance. Here are the results when only the seedling survival life history trait evolves. And to study extinction, we define an arbitrary critical threshold below which we consider the population as extinct because of stochastic events. And it allows us to define minimal genetic variance, so here, below which the population drops below the threshold because the evolution is too slow. And it allows us to define a Vmax, a maximal variance, which is an amount of variance above which the population drops below the threshold too. And this Vmax is due to the variance load. So in a population, when there is variance, some individuals do not have the best phenotype, so the population growth rate decreases. And so Vmax is an amount of variance so high that the variance load causes the decline of the population. So as you can see, you can identify a range of variance allowing rescue. And you can identify this range when each life history trait of the life cycle evolves alone. As you can see, when the flowering rate life history trait evolves alone, the population can't be saved. So I did not represent the result for this trait after that. To compare the range of variance, we can represent all the results on the same figure. And as expected from our analytical prediction, the minimal variance decreases with increasing elasticity. So it is because traits with high elasticity evolve faster and have stronger positive impact on population growth rate. So you need less variance to observe rescue. And concerning Vmax, in reality, it depends of the population growth rate value when the considered life history trait is maximized. So if you consider a life history trait, when this trait is maximized, the population growth rate is really high, the tolerable variance load is higher. Okay, so after that, I considered a multivariate scenario where all the 4 life history traits of the life cycle are genetically variable and evolve simultaneously. So I here represented a bivariate case for simplicity, but so I generated many genetic variance covariance matrices, so you can represent them by an ellipse. And for each one of these matrices, I computed the amount of variance in the direction of the elasticity vector. So the elasticity vector is the vector containing the elasticity of the different life history traits, and it is the direction that maximizes population growth rate. So as you can see, depending on the matrix, you can observe different amounts in the direction of the elasticity vector. It depends on the orientation of the matrix, but also on the total genetic variance it contains. And again, I have analytical predictions, this time in multivariate. So the multivariate change of the mean life history traits is given by the product of the G matrix and of the elasticity vector. And we expect that the higher the amount in the direction of the elasticity vector, the higher the speed of evolution, and thus the higher the probability of rescue. And our results show a strong relation between the amount of variance in this direction and the probability of rescue. Each dot is a G matrix. So, yeah. The more the variance is in this direction, the higher the probability to observe rescue. And if you fit a statistical model, you can also define minimal variance as an amount of variance below which the probability of rescue is less than a given percentage. Here it's 50%. Okay, so to sum up, rescue is more likely when variance is distributed on high elasticity traits. I also showed that there is a range of variance allowing rescue, and the lower bound V mean minimal variance depends mainly on the elasticity of the trait, but also on population size. I did not show these results. And the upper bound V max depends on population growth rate value when the considered life history trait is maximized. I also confirmed these results for individual based simulations. I did not show this result this time. And just one question we could ask is, are these high elasticity life history traits genetically viable in natural populations because they are under strong selection? So to answer this question, we need quantification of the additive genetic variance in natural population. And concerning the Santorea-Corambosa case, in Montpellier, there is an experiment going on, and we will be able to compute the G matrices. And because we have, from the demography, the direction of the elasticity vector, we will be able to assess the probability of rescue of the natural populations. I just want to thank Ophélie Ronce and Olivier Coteau, my supervisors, the ISEM lab I'm working in Montpellier, and the Evo team. And thank you. I'm happy to take some questions. Yes.
Excellent talk. With the limits of Vmax, would tweaking that assumption of constant genetic variance potentially just keep moving that further up as variance will be lost over time?
Yeah, that's exactly what we expect. And I'm running some individual-based simulation where this assumption is relaxed. So, and that's kind of what I'm seeing, yeah.
Because the life history that you're considering has the rosette stage, which
Yeah.
the plants can stay in for a long time, and you can end up decoupling selection from the stage that it's in, right? Because they stay in the same stage for a long period of time. Does your approach allow you to sort of play with the life history itself of the organism to see if how changes in the life history would affect some of your results?
Good question. So, you mean like if
Increasing probability of remaining in rosette stage, so that it's had a much longer lifespan and it takes a lot longer to reach flower.
yeah, so I guess so, because the fact that you stay in the rosette stages depends on two different life history traits. The fact that you don't flower and the fact that you survive. So in certain scenarios, I guess that the life history cycle changes and you either stay longer or either you just pass through 1 year and then flowers. I don't know.
So you were keeping the life history to match the real organism. You weren't varying the life history itself.
When the life history traits evolve, of course it changes the life cycle, because if the flowering rate is really high, you don't stay in the rosette stages. So it changes. Yeah, depending on what life history trait evolve. Yeah.
I was going to ask about the flowering rate. Why did you not get any rescue with the flowering?
Yeah, actually it is because when this trait is maximized and the other ones set to their average value, the population growth rate value is less than 1, and so the population is still declining. Yeah.
All right. Thank you very much, Julien.
Thank you.
Thank you. Thank you for the introduction. There we go. That's what I want. So, as was just said, I'm Jeremy Summers. I'm a graduate student at Nancy Chen's lab, and today I'm going to be sharing with you some really exciting results that we have looking at the multi-generational impact of gene flow in a natural population. So with some of these lovely talks that we've been looking at, we've been considering evolutionary rescue with new mutations arriving in populations that are relatively large in size. When we're thinking about populations that are small and isolated, gene flow becomes an extremely important parameter to consider how fitness is going to change over time. This is particularly due to some of the fitness benefits that can arrive in these small, fragmented populations through gene flow. Gene flow is going to be maintaining genetic variation to maintain that adaptability and counter variation loss due to genetic drift, and it will also decrease the amount of inbreeding depression that the population experiences. And that's due to the masking the decrease in fitness that arrives with more inbred individuals. Collectively, that's referred to then as genetic rescue. But when we're also considering small populations, we need to be wary of potential outbreeding depression. This can be caused due to countering local adaptation between multiple populations with different fitness peaks at different locations, and also the breakup of co-adaptive gene complexes. There's also the possibility of introducing deleterious variation. Different populations can have different levels of deleterious variation, and thus gene flow can potentially influence populations in that way as well. However, these changes in fitness are unlikely to remain constant over time due to differences among individuals. So if we consider a resident individual who's from a local population and a new immigrant, when they pair, they would produce an offspring that we could call an F1. And they would have one copy of their genome from their resident parent and one copy from their immigrant parent. Thus, they are going to be heterozygous at any site where there are differences between the resident and immigrant populations. If this F1 were to then pair with another F1, they would produce an offspring, an F2, who still has half their genome representative on average from the resident population and half from the immigrant population. However, due to recombination, they can now be homozygous for resident or immigrant alleles at different locations along the genome. And similar amount of variance can occur with various different types of crosses. F1s could pair with residents and produce resident back crosses. They could pair with immigrants, produce immigrant back crosses. And if there's a sufficient number of immigrants in a population, they could even reproduce on the population and produce locally born individuals of entirely immigrant descent, here referred to as immigrant offspring. And so there's different expectations that we'd have for how fitness would change across those different groups, depending on what underpins differences in fitness between immigrants and residents. If it's primarily driven by additive effects, where the amount of immigrant ancestry could perhaps additively increase fitness, so the F1 individual who's heterozygous is intermediate between immigrant and resident fitness, then we would expect that individuals with greater immigrant ancestry would have the maximum amount of fitness within the population, and that would vary continuously between these different groups. Alternatively, if it's primarily driven by dominant recessive genetic effects, so this is mostly looking at alleles where the phenotype is going to match one or the other parent depending on dominant or recessive alleles, then that follows more into the classic inbreeding depression framework, where a heterozygous individual may mask deleterious recessive variants due to being heterozygous and carrying a dominant allele from one other parent population. And in that situation, the most heterozygous group, the F1 individuals, would have the greatest relative fitness within the population. But we also talked about co-adaptive gene complexes, epistatic effects, interactions between different loci on the genome. Within that situation, which would largely be driven by local adaptation, those F2 individuals that can be homozygous at different locations, they may incur a fitness cost due to the recombination of their genomes, breaking up those local co-adaptive gene complexes. But then, of course, none of these genetic effects could play a role as well. There can be ecological or behavioral effects that differentiate immigrants from residents. Within these social or environmental aspects, we'd probably expect that new immigrants into the population would have different fitness from all individuals. Perhaps they could be doing much better or much worse, depending on familiarity with the environment or relatedness between individuals. And there hasn't been a lot of explorations of multi-generational fitness in natural populations. But recently this year, an excellent study from Dickel et al. Came out that was looking at a population of song sparrows in the Pacific Northwest. This population had a relatively low immigration rate, but what they were able to find was that specifically among males, there was a difference in the F1, so that second row there is the difference between resident individuals and F1s in lifetime reproductive success. There was also a difference in F2 males, which was negative, which matches those expectations of both that masking of deleterious variation with inbreeding depression, but also those epistatic effects, a bit of outbreeding depression occurring as well. But we really need to find more studies that look at multi-generational fitness in immigrants, and especially in systems with higher immigration rates. Frequently, conservation goals are to expand connectivity and gene flow across populations, so populations with high immigration rates are sort of the end goal of those conservation efforts. Luckily, there is an excellent population that can provide some data on this. There's a population of the Florida scrub jay. This is a species that is federally threatened. It's only endemic to the state of Florida, and it's threatened mostly due to the loss of habitat. That gray area is the historic range of the species across the state, and it now exists in small, patchy habitats. They are also cooperative breeders, meaning that individuals will stay on their natal territory potentially for several years and assist their parents in rearing young. They will also potentially disperse to neighboring territories and assist there as well. So these individuals stay on these territories year-round and interact with each other. So there is plenty of space for social factors to potentially influence immigrant fitness. And then finally, with this specific population at Archibald Biological Station, we have more than 50 years of census and fitness data and a multi-generational population pedigree. So we have the data to identify immigrants, residents, and their descendants over several generations. What makes this population especially interesting, in my opinion, is that it is also an extremely well-preserved population, while surrounding populations are rapidly declining. And that mainly manifests in a decline in the immigration rate over the past 30 years, going from an extremely high immigration rate, above 60 individuals per year, now to a more moderate, but still relatively high compared to many populations. And what's coincided with that decline in immigration has been an increase in average inbreeding coefficients among breeders that has rapidly spiked very recently. So to actually investigate this, what we really need to first do is categorize individuals across those different immigrant ancestry categories, which we're able to do over years from 1995 all the way up to 2023. And you can even see with these different categories, those bluer groups are those individuals of more immigrant ancestry. They are declining over time, which matches what we would expect with a decline in immigration. So with our categories, we then want to create Bayesian generalized linear mixed effect models that look for differences in fitness metrics between each of those groups and resident individuals. The fitness metrics that we ended up looking at was juvenile survival, which is survival from 11 days of age, which is when we put a band on them and start monitoring those individuals, to their next breeding season. And then also adult survival, survival for each annual year as an adult, and likelihood of establishing as a breeder at some point. And then finally, there's annual reproductive success, which finishes the Florida scrub jay life cycle. Overall, that becomes lifetime reproductive success, but we can also run a model separately that looks specifically at breeder lifetime reproductive success. So let's go into these results now. Starting out with juvenile survival, to get you used to all these plots, there's going to be a few of them. These are the posterior differences between each of these different ancestry groups and resident individuals. So a difference over here that we have with immigrant backcross would indicate that there is about 10 percentage points increase on the mean estimate for juvenile survival in females. However, the credibility interval that we have indicates that's not a significant difference. In fact, across juvenile survival, we don't see any of these differences. But if we then move on to adult annual survival, then we do find that there is a significant increase in immigrant backcross individuals, specifically females, with their survival, which would indicate that, and there's also this positive trend among immigrant offspring and immigrant individuals themselves, which does seem to line up with that increased immigrant ancestry increasing fitness. When we look to transition to breeder, the main strong effect that we indicate is that immigrant females are much more likely to establish as a breeder. They will become a breeder much sooner than a resident-born individual. Though there is also this slight support for a decrease among immigrant offspring with males, and also with resident backcross males. With annual reproductive success, females, though, do not show any signs and differences in fitness, but instead with males, we see that decline in F2, which matches up with what the song sparrow study found as well. And then finishing this all up, taking it together for lifetime reproductive success, we find that there is overall an increase in lifetime reproductive success among immigrant backcross females, and some support also for new immigrant females, with positive trend as well with immigrant offspring. So that's lining up with that idea of increased immigrant ancestry, increasing fitness. Putting this all together, that increased immigrant ancestry, increasing fitness, especially among immigrant backcross females who are resident-born individuals but have a large proportion of immigrant ancestry, would support the idea of additive effects potentially factoring into this system. So there could be new beneficial variation being introduced from immigrants. Whereas we do also find a similar result to the song sparrows with declines in annual reproductive success in males among F2 individuals. And then finally, we do also find an increase in female transition rates, though that likely has a large effect on the ecological or behavioral effects, because we see it primarily in new immigrant females. And also the likelihood to establish as a breeder is going to involve a lot of different social effects as well. And in fact, some specific details that would probably clear some of this up as well is that there is female biased dispersal in this system. So females are more likely to disperse into the system and more likely to disperse out as well. Male fitness is more tied to the ability to inherit territory, whereas female fitness may be tied more likely to moving to a new territory and establishing as a breeder. But there could also be variations in the heritable variation in fitness that is different between males and females. In fact, overall, I would say our results line up with what was previously found in those song sparrows, that there are sex-specific impacts of gene flow, and that there's potentially differences in heritable variation among males and females. We do find that there's a fitness benefit of gene flow even when there is extremely high immigration, which is good news that maintaining connectivity within populations can still maintain the benefits of gene flow. And then we also find evidence of both genetic and social factors that can influence immigrant fitness in a species with a complex life cycle. Some future directions that we're probably going to end up exploring is looking at differences among immigrants. We want to note that immigrants can come from many different populations, and they may be more or less related to residents. We're looking to do some whole genome sequencing results to explore genetic structure among immigrants and their relatedness to individuals. This is a photo of me shipping out our last box of samples of this 4,000 individual study that we are putting together for this earlier this month, so hopefully some more results soon. With that, I would like to acknowledge everyone who has made this project possible, especially all the people who work so hard at Archibald Biological Station and my various funding sources and members of the Chen Lab who have helped me along the way. With that, I would be happy to take any sort of questions, and also want to make a note that I'm currently looking for postdoc opportunities, especially interested in looking at time series of genomic data. So thank you for listening to this. Happy to take any questions. Yes.
So it's interesting that the immigrant females have an ecological advantage because it's not completely intuitive in a cooperative breeding species. So do you think it's because of behavioral syndromes, like immigrants are more aggressive, or like why do you think they establish that?
That is an excellent question. A very exciting thing that we've been wanting to look at is comparing some of the different behaviors between immigrants and residents. Some factors that likely play a role with it, possibly, is that these immigrant females are much less related to potential mates within the population relative to resident females, and that within the social hierarchy of the Florida scrub jay, male individuals tend to be more dominant with it as well. So there may be a better ability for these less related females to establish as breeders if the resident males are more controlling the breeding opportunities within there. But there could definitely be some differences, especially after the selection of dispersal. Yes?
I know you guys have tons of data on the amount of inbreeding for specific individuals within that population.
Yes.
And so you can have a sense of how inbred they are. Do you have any sense of how inbred your immigrants are?
Yes, so there's been some previous work done that's done some B-chip data. So we have some selection of SNPs to be able to compare this. And immigrants in general are more inbred than resident individuals, which lines up with our understanding that they are probably coming from much smaller populations that are actively declining. Oh, yes, all the way in the back.
Yeah, well, that's beautiful book, thank you. I was just wondering, so you've got kind of categorical thing of features and so on, but we know the realized ancestry in those is different, right? So I just wondered if you get more out of that, or if you're trying to continuous measure of ancestry, you're already one already.
Yes, thank you. Excellent question. So that question is if we looked at more continuous measures of ancestry rather than using these categorical variables. We have looked at specifically like number of immigrant ancestors, so we can count the number of immigrant grandparents this individual has, and we're working on getting that genetic data for the entire study site so we can actually differentiate it and say, you know, is this immigrant more or less related to residents? How closely related are you to more distantly related immigrants? So that's something that we're working on. With the number of immigrant ancestors, we see essentially the same results arriving within this, especially with the increased immigrant ancestry in females. Thank you.
Thank you. Okay, thanks for coming today. So I'm going to talk about specialization, niche partitioning, as they can play a role in evolutionary rescue. So this is kind of preliminary work. This is a single author paper so far. So I do want to thank members of my lab. We do create that intellectual environment for each other, and they've certainly contributed. And I want to mention my past advisors because I think all of them have influenced this particular work and, of course, funding sources. So we often talk about, does evolutionary rescue happen or not? I want to think about what happens next. Does evolutionary rescue change a population? How does it do so? So how is fitness restored if there's an abrupt environmental change? When we look at natural examples of evolutionary rescue, such as the silent crickets in Hawaii, we see very substantial, that's going to happen a lot, very substantial changes that might ripple through the life history of that population, have all sorts of demographic effects. So we don't simply see a restoration to what was there before. Similarly, when we think about moving optima models where populations are tracking a moving optima, simple predictions can, we can kind of come up with an expectation that they may do so indefinitely, but we know things can change. We know populations might run out of genetic variants, they might hit certain hard limits as to how much traits can move, but in the opposite direction, populations, the evolvability of populations can change, can respond to the need to adapt in various ways. So can evolvability evolve to help populations track environments? Yes. We've already seen examples of that today in the form of mutation rate. Recombination rate is another well-known example. We have examples of this, particularly in models, with looking at genetic architecture. But what I want to talk about today is niche breadth and the role it can play in evolvability. So adaptation in generalists, by which I just mean a species or a population that's using multiple environments, can be constrained in several different ways. Commonly, we think of them as constrained by trade-offs. The same allele is not good in all those different environments. However, even without those kind of trade-offs, generalists suffer lower evolvability, and there's a nice literature looking at this in a bunch of different ways. However, we don't have a lot of examples of how this could play out in an evolutionary rescue scenario. So one really standout theory paper looks at this in the situation of antagonistic coevolution and asks, is the higher evolvability of specialists allow them to dominate and displace a generalist in a situation with antagonistic coevolution in a red queen scenario? So this is a really awesome paper, but I think we need to continue to look down this road for a few different reasons. One is simply antagonistic coevolution is too complicated for me to understand. We want to maybe back out of that scenario and look at a situation where we can control the rate of environmental change a little bit more as an independent variable. We know that specialists can suffer ecological costs, particular costs of having to search for longer to find their preferred habitats. They may also have a smaller population size because they're only using a smaller portion of a portfolio of possible environments. We want to get that factor into this model too. And we also want to focus on the role of mutation. So in this Kvecki paper, specialists and generalists are already present in a deterministic framework where extinction doesn't happen. But could we get specialist mutants that arise in a situation where they have evolvability benefits, perhaps, but how would that play out? If there's short-term costs on those mutations, but long-term benefits, those long-term benefits may never get realized if those mutations go extinct. So even if we might hypothesize that specialists are optimal, how do you get there from generalists, given natural selection? So my approach to this is to look at a population that's a generalist population spread across M different environments. Juveniles are going to visit sites until they pick one or die, and that's how we're going to impose search costs, is that if you're picky, you have a higher chance of dying before you find something that you like. And particularly specialist genotypes are going to be choosy, so they're going to reject habitats. And fitness in each environment is going to be determined by its own independent set of loci. So this is not necessarily a realistic choice. It's made in order to ensure that there's no antagonistic pleiotropy. There's no genetic trade-offs which force you to be a specialist versus a generalist. You can be perfectly adapted to everything if you can keep that, if you can keep up with environmental change. So an example genotypes, in this case, let's say we have 5 loci for each of 4 environments, and each of them has 26 different alleles at each of those loci. So we could compare that to optimal patterns, which basically just describe what would be the best genotype to have in each different environment, and impose fitness costs for mismatches based on how you're matching that pattern. And finally, we can also have loci that track your preferences. So in this case, we'd be looking at an organism that would happily settle in environments 1, 2, and 4, but would reject environment 3. So all these things are mutable. And what I'm going to show you today is some pretty preliminary results from simulations. Not going to try to digest this too much. So that's the stage of the project. So our question is a little bit different from what we may have seen in some abrupt change models of evolutionary rescue. We want to know, can populations survive a period in which environments are changing rapidly? So in other words, I'm going to turn up the rate of environmental change itself to potentially unsustainable levels and see what happens. So here's some examples of a situation where preferences can't evolve. And I've just basically run ensembles for each of 3 different rates and split them into subsets that do and don't survive. So for example, at this highest rate of change, none of our populations survive. At the lowest rate, over half survive, maybe about a third go extinct. In the middle, it's about 50-50. So if we allow preferences to evolve, we see something different, which I'm calling evolutionary rescue, at least in a statistical sense, because we see now at this highest rate of change, now some of the populations can survive. In general, we see more survival. However, we don't necessarily see all the signatures that we typically associate with evolutionary rescue. We see that our populations stop that sort of slide into extinction, but they don't necessarily bounce back. They're keeping up with environmental change. They're not recovering in abundance. There's also another reason they're not recovering in abundance, because now they have a smaller niche, so they have a smaller total population size. So abundance is stabilized but don't recover in this model. And we can look at this more broadly and see that as we increase that rate or the probability per unit time of environmental change, we get to unsustainable rates of change. And if we allow preferences to evolve, we see a much broader range of parameter space where populations can persist. So this is what I'm calling evolutionary rescue here. Let's look a little bit about how this happens, what this has to do with niche partitioning. Okay, so here's just a single example, and it's a bit complicated here, but basically we're seeing decline in fitness in 4 different environments, color-coded here. And then on the bottom, we see all sorts of different preference patterns, and this is just looking at a case where preferences are binary. As you can see, that still produces quite a bit of complexity here. And what we see is the generalist eventually goes extinct, and if we fast forward to the end, we end up with 2 surviving lineages that are narrow specialists, and then we have 2 niches which are unoccupied, the blue and the purple. So no one ends up using those at the end of our simulation. And we can start to see a little glimmer of what might be going on, how mutations can be beneficial in this situation. So if we look, actually the first thing that really takes off here is a genotype that uses everything except the cyan environment here. The environments aren't any different from each other for any intrinsic reason, but we can see that early on, fitness in that cyan environment falls first. So that's probably just a stochastic product of a little bit slower adaptation rate in that environment, a little bit faster environmental change. But what that means is that I suspect that that mutation, which narrowed the host range, was actually beneficial in the moment as well as contributing greater evolvability. And why was it beneficial? Because of that little stochastic difference in the fitness of those environments that opened up. This is an anecdotal result so far that I hope to build on. We can look at a few other things too. So we can see that as we ramp up to higher and higher rates of environmental change, the number of environments that are used by our surviving genotypes goes down. So at Intermediate rates, we may see genotypes that use 2 out of 4 environments towards these much higher rates that are only barely possible for our populations to survive. They narrow even further to the highest level of specialization. If we pick one of these particular treatment conditions and look at possible end products that we get across replicate simulations, we see a pattern that looks a lot like genetic trade-offs are at play. We see that the broader your niche is, the lower your fitness is, averaged across all those environments in your niche. So if we saw this pattern empirically, we would probably interpret that as antagonistic pleiotropy. We would interpret that as genetic constraints on adaptation. But in fact, it's purely the product of these evolvability constraints on niche breadth instead. Finally, we can relax the rate of environmental change after this treatment is over. We see that fitnesses bounce back up once that really high rate of environmental change is no longer present. Then we can simply ask, do populations then kind of restore back to a single generalist dominating the environment? And what we find, I tried to make a figure, but the answer is no, they don't change at all, so the figure is boring. The number of rejected environments, the niche breadth, doesn't change when we do that, at least for periods of time that are kind of amenable to simulation. So what we see is pretty long-term impacts on the niche, even after that period of accelerated environmental change is over. Okay, so take home so far, regimes with faster changes evoke greater specialization and leave abandoned parts of the environment. So abandoned sort of parts of niche space, in this, at least in this asexual framework that I'm looking at so far. Trade-offs between niche breadth and evolvability can produce patterns that look like genetic trade-offs, and niche contraction can be very hard to reverse. Okay, so next steps, focus on the fitness of those specialist mutants to answer the question, do these mutations that enhance evolvability, are they also beneficial in the moment? And particularly look at differences between asexual and sexual evolution. We certainly expect a lot of differences here because we expect sexual populations are going to be much better at adapting to multiple things at the same time. Finally, of course, when are these evolvability differences that we're dealing with hypothetically here, where could these be relevant in nature? Okay, that's all today. Thank you very much. Fred. Oh, I'm sorry.
You leave.
Okay.
Hi. So, yeah, very interesting talk. As you drop from 4 to 3 to 2 to 1 environments being exploited, do you think that's just on the road to going extinct altogether if you ran the model for longer?
Yeah, so that's a great question. I think it's fairly stable. We do, so what we're defining as rescue is surviving for a finite period of time, of course. So I haven't rigorously looked to make sure that everything that I'm calling a survivor, you know, would most of those stick around if we say doubled that period? I suspect that's the case, but I need to do more to follow that up.
Do you think that we can explain some of the results by competitive exclusion between different types, or are there no demographic effects here?
There's definitely demographic effects, and absolutely, I think I would like to be able to do that. I can't explain it today. Yeah, I think this model of the way demography works in this, I intended to make it pretty simple to analyze that. I'm not sure if I actually achieved that, though. So I'm hoping to do that, yeah.